video on sensation. So first, sensory receptors. Some of them will combine with muscles and tissue to form sense organs. Most of these will consist of specialized nerve cells or nerve endings. And we're going to talk about some of the specific types in a second. So some of the information they transmit includes the type of stimulus, the location of stimulus, and the intensity of stimulus, such as the loudness of a sound. We can classify receptors by the type of thing that they detect. So chemoreceptors detect chemicals. Examples would be smell or taste or pH. These use chemoreceptors. Mechanoreceptors are used for touch, pressure, stretch, vibration. Then thermoreceptors detect hot or cold, changes in temperature. Nociceptors are for pain. Photoreceptors are for light, found in the eyes. Proprioceptors detect muscle stretch and body orientation. They tell you where you are in space, and they allow you to move in a coordinated fashion. They're going to send information to your cerebellum and help you keep your movements coordinated. Alright, next thing is sensory adaptation. So your receptors are specialized to detect change, because a change might be a threat to you, such as a bear. So if a stimulus is constant, your body adapts to it and ignores it. So for example, you get into cold water. At first it's super cold, but then your receptors sort of adapt, and after a while you don't feel it as much. And smell is our fastest adapting sense. Alright, a question nociceptors respond to, and the correct answer is pain. Alright, the general senses include pain, pressure, touch, stretch, and temperature. These are found in receptors. These receptors are found in the skin, so pretty much all over the body. Nociceptors consist of free nerve endings that carry impulses to the brain. These receptors are abundant in the skin and mucous membranes and in almost every organ. They use two types, fast and slow pain fibers. This shows pain pathway. We're going to watch this animation.
When an injury occurs, injured tissues release chemicals that stimulate nociceptors and trigger pain. A neuron conducts a pain signal to the dorsal horn of the spinal cord. At the dorsal horn, the spinothalamic tract relays the pain signal to the thalamus. At the same time, the spinal-reticular tract carries pain signals to the reticular formation of the brainstem. The thalamus relays the signal from the spinothalamic tract to the postcentral gyrus of the cerebrum. At that point, the person becomes aware of the pain. Meanwhile, the impulse from the spinal-reticular tract bypasses the thalamus and travels to the hypothalamus and limbic system. These areas trigger emotional and behavioral responses to pain, such as fear and nausea.
Alright, so to review, the injured tissues are going to release chemicals that stimulate the nociceptors. The sensory neuron is going to send that signal to the spinal cord. Then in the spinal cord, it's going to go kind of two places at the same time. The signal is going to be relayed to the thalamus. They're also going to be sent to the brainstem, to the reticular formation. Then the thalamus is going to relay that signal to the cerebrum. And that's when you become aware of the pain. And at the same time, the signal is sent to the hypothalamus and the limbic system. These control emotional responses like fear and nausea. Alright, next thing is referred pain. This figure shows some examples of referred pain. One that probably everyone has heard of is your heart. So if you have pain on the left side of your chest and your left arm, it could be a bad sign of a heart attack. And referred pain happens when sensory pathways from an organ and an area of skin converge in a single pathway, and then there's kind of our nerves get a little confused. And so for example, if there's pain in your heart, that gets carried in the same pathway as the pain from your left arm, and so your body gets a little confused. Alright, why do we have pain? Most people don't like pain. However, it alerts us to danger, and it lets us know there is an injury. You might think it would be nice to not have pain. There's a disease called congenital analgesia, where people don't feel pain. It's becoming more treatable now that we're aware of it, but these people have to be super careful because they might, for example, cut themselves and not realize it and then bleed to death, or they might burn themselves and get an infection and they don't even know they have an infection. Like, they could have a cavity in their tooth and not know it and get an infection that way. So we actually need to be grateful for pain. Alright, the special senses are taste, smell, hearing, equilibrium or balance, and vision. These use specialized organs. These are all found in the head. And we're going to look first at the sense of taste. So in our mouth, inside our tongues, we have taste buds, and the bumps you see on your tongue are not actually taste buds. The taste buds are microscopic. They're down sort of in the folds of these taste buds. And if we take a closer look at a taste bud, we'll see they have these little hairs. So when chemicals come in contact with those, it activates these cells and they send a signal through the nerve to the brain. That's how taste happens. Next thing is the 5 tastes. So salty, sweet, sour, and bitter, most people are familiar with. Umami is a newer one. I call it the Chinese food taste. It's been described as meaty or savory. It's from a Japanese word that means delicious. Alright, next is the sense of smell. So the way the sense of smell works, up inside your nasal cavity you have these little nerves coming down, and odor molecules will enter your nose and bind with one of these receptors. These receptors send a nerve impulse up to the olfactory bulb in the brain, and then that's going to travel to the primary olfactory cortex, where it then gets converted to smell. Smell and taste are related. Taste is about 60 to 70% smell. That's why, for example, if your nose is stuffed up, you can't taste very well. So if you have to eat something that you don't like the taste of it, just plug your nose and you won't smell it as much. Alright, why do certain odors invoke a memory? So homemade bread might bring back a memory of your grandma's house, or a certain perfume might bring back a certain memory. It's because smell information, as well as going to the olfactory cortex, it goes to the hippocampus and amygdala. There it can evoke a memory, an emotional response, or even a physical response such as vomiting. Alright, next we're going to look at some parts of the ear. So let's start with the different parts. So on the outside we have the auricle. That's the part you can see. It funnels sounds into the auditory canal. Then the auditory canal is kind of just a passageway for those sound waves to travel. Sound travels as a pressure wave. The auditory ossicles are the ear bones you can see in the middle there. They basically transmit the vibrations, so the sound travels through the auditory canal, then the tympanic membrane vibrates that, and those vibrate the auditory ossicles, so those are transmitting the vibrations. And then the oval window, in this figure we can see the close-up on the middle ear and the oval window. So the stapes is the third ear bone. It moves against the oval window and vibrates the fluid in the inner ear. Also here we can see the tympanic membrane. The tympanic membrane vibrates in response to sound waves. Also here we can see the eustachian tube or auditory tube. Its job is to equalize the pressure between the inner ear and the outer ear. It goes all the way down to your throat. The The semicircular canals are involved in balance, and we can see also the vestibule also involved in balance, and then this thing that looks sort of like a snail shell is the cochlea. It's involved in hearing. And if you take a close-up look at the cochlea, you'll see inside the organ of Corti, and this tectorial membrane actually vibrates, and then those what are called hair cells in there, they become activated, and that's how we detect sound. So why are middle ear infections more common in children? Because in children the auditory tube is short and pretty much horizontal, so drainage from the nose can flow to the ear, and that can spread an infection. And as you grow, the tube's going to get longer, and it's going to start to slant downward, so it's harder for drainage from the nose to get to your ear, because it has to go against gravity. Alright, let's watch this animation on how hearing occurs.
Hearing occurs when sound creates vibrations in the air, known as sound waves. Sound waves enter the ear and travel down the external auditory canal. The waves strike the tympanic membrane, causing it to vibrate. The vibration spreads through the malleus, the incus, and the stapes, the three bones of the middle ear. The movement of the stapes against the oval window shakes the perilymph on either side of the cochlear duct. The ripples in the perilymph are transmitted through the roof of the cochlear duct to the organ of Corti. Movement here stimulates the hairs of the organ of Corti to send nerve impulses along the cochlear nerve. The impulses reach the auditory cortex in the brain's temporal lobe, where it is interpreted as sound. The ripples continue through the perilymph and dissipate by striking the round window.
Alright, so just to review, the sound comes in, vibrates the tympanic membrane, and those vibrations travel through the auditory ossicles. The oval window is going to push against the cochlea and cause vibrations in this fluid. That's going to vibrate the organ of Corti and cause action potentials to be generated, and then those vibrations leave through the round window. That way your eardrum doesn't explode, or your cochlea doesn't explode. So again, sound waves travel down the external auditory canal and vibrate the tympanic membrane. Next step, vibrations travel through the malleus, incus, and stapes. Then the movement of the stapes against the oval window shakes the fluid in the cochlea. Then fourth, the vibrations travel through the fluid to the organ of Corti. This sends nerve impulses to the brain, where it is interpreted as sound. And then last, the vibrations leave the cochlea through the round window. So, which inner structure contains the structures for hearing? And the correct answer is the cochlea. Alright, there are two types of hearing loss. First is conductive. This is problems with transmitting sound to the inner ear. So it's a problem in the outer or the middle ear. Sometimes those can be surgically corrected. Sensorineural hearing happens with death of hair cells in the organ of Corti, and these are never going to grow back their neurons, so they don't regenerate. Now we have some pretty cool technology. We can fix this with a cochlear implant. Alright, next we're going to talk about balance. So in the ear, it's hearing and also balance, and so we have these semicircular canals, and they're oriented in the three different planes, so they can detect angular motion in all three directions. So if we look inside, if we look at the base of each semicircular canal, there's an ampulla, and if we look at a close-up on the ampulla, there's this cupula with hair cells. And as we rotate, for example, as you turn your head, the fluid inside the semicircular canal is going to move, and that's going to move this cupula, and it bends those hair cells right there. And as those hair cells bend, then they send signals to our brain to let us know our head is turning. If we look at the vestibule, inside the vestibule, we have these otoliths. They're like crystals, and they're over other hair cells. And they're more involved with detecting static equilibrium. So, for example, if you bend forward, these otoliths are going to move forward, and that's going to bend the hair cells. These are also going to be involved in detecting linear acceleration. So if you move forward, those otoliths are going to move back and stretch those hair cells. And so we need both of these together to give us our sense of balance. And the utricle and saccule reside inside the vestibule. Alright, next we're going to talk about vision. So let's start with parts of the eye. The eyelid is on the outside is for protection. These close so we can sleep. The eyelashes are also for protection. They keep foreign things out of our eyes. The conjunctiva is a thin layer around the eye and the inside of the eyelids. It makes mucus. It keeps the eyeball moist. Then in the next figure we can see the lacrimal gland. It makes tears, which helps clean our eyes. We can also see here the nasolacrimal duct. This carries tears down to the nasal cavity, so that explains why you get the sniffles when you're crying. Then we're going to look at the eye. The sclera is the white part on the, it's the outermost part of the eye. It's the whites of your eye. The cornea is kind of continuous with the sclera, but the cornea is clear. That's the front part of the eye. So it lets light in and it does a little bit of focusing. The iris is the colored part of your eye. It adjusts the diameter of the pupil, so it controls how much light is going in. Then the ciliary body we can see down here, it secretes the aqueous humor, which is the fluid that is found in the front part of your eye. The choroid is like the middle layer of your eye. The choroid contains blood vessels to nourish the other layers. Then the retina is the inner layer, and it contains the photoreceptors, so that's where we actually see. So in this figure, the retina is actually yellow. And if we take a closer look at the retina, actually, I'm going to do the optic nerve first. So all the photoreceptors in the retina send their signals out, and they combine and they leave here and form the optic nerve, which then goes back to your brain. So it sends visual signals to the brain. Then if we look at the retina, we see in the center, the macula lutea is the center of the retina. It has lots of cones, so it gives us a sharper vision. Then in the very center you have the fovea centralis. It's an area with really, really concentrated cones, just cones. That's where we get our sharpest vision. And when we're looking at something, we want that light to be directed at our fovea. That's why, for example, when you're reading a book, you would move your eyes along the page so that whatever you're trying to read is focused in your fovea. Then the optic disc is off to the side. It's where all the nerves leave the retina. It's also where blood vessels enter and leave. So the outermost layer of the eye is the sclera. And if we look at a cross-section through the eye, we can see the anterior cavity. This is between the lens and the cornea, and this layer is full of aqueous humor. You can see these arrows showing where the aqueous humor is produced, and then normally it's reabsorbed there in the canal of Schlemm, so we're constantly making more and reabsorbing it. Also here we can see the lens. The lens, its job is to focus light. And then the posterior cavity is basically the back of the eyeball. It's full of the vitreous humor, which is more like jelly. Alright, next thing is glaucoma. So we mentioned the aqueous humor is normally being produced and reabsorbed, but if that canal of Schlemm that we see here, if that gets blocked, then the aqueous humor will build up pressure in the anterior part of the eye, and then that will cause pressure in the posterior part of the eye. The vitreous humor can press against the choroid, and then that blocks the blood flow, and if these photoreceptors don't get oxygen, they die. So this can cause blindness, although it can be treated with medications. Alright, so let's talk about the process of vision, the things that have to happen for vision to occur. First, light focuses on the retina and produces an upside down image. The rods and cones then convert the image to nerve impulses, and then those impulses are transmitted to the brain. So here we can see light being sent to the retina, where it would actually be upside down on the retina, and then again the rods and cones convert the image to nerve impulses, and then those are transported back to the brain. Alright, next we're going to look at some of the common visual defects. So myopia or nearsightedness, this is when it's actually hard to see things that are farther away. And you can see the light is being focused in front of the retina instead of on the retina. With hyperopia or farsightedness, the light is focused behind the retina, so with this it's harder to see things that are close up. Astigmatism results from an uneven or asymmetrical curvature of the cornea, so you can see the light is not focused in the right place. These first three, those can all be corrected with glasses or contacts very easily. Next one is presbyopia, so with age the lens loses flexibility and it gets harder to focus on things that are close up. That's why as you get older you have to hold papers farther away from your eyes to be able to focus on them. And then the last one is cataracts. So you can see a picture of someone with cataracts. This is when the lens gets cloudy and it's hard to see through that. Cataract surgery is pretty common now. They can put an artificial lens in and restore your sight. Alright, next thing is the photopupillary reflex. So there are two different muscles that control the pupil. The pupillary constrictor is going to cause it to get smaller to let less light in. The pupillary dilator is going to make the pupil bigger to let more light in. So if you're out at night, you want more light to come in. The photopupillary reflex is activated when you see bright light. So for example, when you go out into the sun, it's going to make your pupil constrict, get smaller, so less light comes in, because too much light can damage your photoreceptors. So for example, if you're staring at the sun, that's going to damage your photoreceptors. So this lets less light in so your photoreceptors will not get damaged. Alright, next thing we're going to compare rods and cones. So rods are found mostly in the periphery of the retina, so on the outside, and cones are found mainly in the center of the retina. Rods are active in very dim light. It doesn't take a lot of light to activate them, they're more sensitive. Cones are active in bright light, so it takes brighter light to activate them. Rods are responsible for night vision, or any vision in low light, and cones are responsible for sharp vision and also color vision. So you'll notice in your peripheral vision, where it's mostly rods, your vision is not as sharp and you don't see color as well. There are some cones out there, so you do see color. And if you go out at night where there's not a lot of light, you'll notice that you don't see colors because there's not enough light to activate your cones. Alright, this shows how we see. So the rods and cones turn the light into nerve impulses, and then they send it back through the optic nerve, which crosses at the optic chiasm. Some of the information is going to go to both sides of your brain, and then in the visual cortex, which is back in the occipital lobe, it's going to be interpreted. Alright, next thing is color blindness. Color blindness results when one or more of the chemicals called photopigments sensitive to a particular color's wavelength are missing. It doesn't mean that the people sees everything in shades of gray. Usually they see color, but they can't distinguish certain colors, and the most common one is red-green color blindness, and it affects men more than women because it is a sex-linked gene. Alright, as we age, there are changes to our body. It is a merciful thing. We're not meant to live forever. But some of the changes with vision, the lens becomes more rigid, making it harder to see nearby objects. The pupil gets smaller, which makes it harder to see in low light. With hearing, the eardrum becomes less elastic, and that causes hearing loss, especially with high frequency sounds. Taste and smell are both going to diminish with age. That makes eating not as fun, might make you lose your appetite. And with touch, the skin sensitivity decreases, making it difficult to identify changes in temperature.